In our final installment for Chapter 10 on photosynthesis, so this makes this Part D, we're going to talk about alternative ways of fixing carbon that will allow plants to survive in times of stress or with given species being able to survive in very, very harsh climates. One of those is going to be photorespiration, which is a common one for most plants, but we're going to talk about some other ones as well. Because plants do depend on water, not only for photosynthesis, but also for relative rigidity and patency of the cells, dehydration becomes a significant problem for plants. So we have to have some trade-offs at some times. That metabolic processes just can't run. They have to be able to turn down or seek alternative outcomes during times of stress. And photosynthesis is certainly no exception because it directly depends on water. So when we're dry, the plants will close their stomata, which prevents water loss, but it also limits photosynthesis. The other problem is that you're going to not have CO2 and you're going to get a rise in O2, which in an animal cell would be just fine, but O2 is actually reactive and is actually a waste product in plants. So we need to get rid of it just like we would need to get rid of CO2 in our bodies. So there are one mechanism that's quite common in most of the plants that you see. And this process is called photorespiration. And so for most C3 plants that we see in and around our lives, what we do is we use Rubisco to fix CO2 to form that 3Pg, as I mentioned in the Calvin cycle. 3Pg then gets reduced to G3P, and that's what we harvest. But in the case of photorespiration, when the plant's under stress, instead of using CO2, Rubisco actually can add oxygen, and instead of creating a 3-carbon compound, it creates a 2-carbon compound because there's no carbon in oxygen, so there's no carbon to add when we use oxygen. So it remains a 2-carbon compound, and then that allows for the consumption of oxygen and the sustainment of the electron transport without really producing any ATP or sugar in the process. It's sort of an idling phase, if you will, in photosynthesis that will prevent that O2 from building up, which can be toxic. By the way, it can even be toxic in animals. Did you know that if you went scuba diving with a tank of pure oxygen, you get down below 30 feet, you will die of oxygen toxicity. And that can occur in plants too. So what do? We do photorespiration in order to get that O2 down, but allow the cycling of electrons to continue. There are a couple of hypotheses bouncing around about what the role of photorespiration is, why we have it. Certainly as a stress protector, perhaps, but there's another hypothesis that says it may just be something that's left over from the time when Rubisco was first evolving and becoming a part of the photosynthetic process. That back then you would have had a lot less O2, a lot more CO2 and other types of gases. There is a hypothesis that photorespiration is a way of protecting cells. That may be why it's stuck around and hasn't been weeded out of the gene pool, if you know what I mean. That when we can't run the Calvin cycle, this allows for continued electron circulation. But the problem is that photorespiration is costly. Because on a hot, dry day, it may drain 50% of the carbon that the Calvin cycle might have produced on a better day, and that's not sustainable. Plants can certainly generate a lot of carbon fixation during a normal growing season, but 50%, that's pretty heavy strain, especially when you consider trying to survive wintertime and that sort of thing. We have photorespiration, and now let's look at ways that plants can do things to minimize the damage that photorespiration may cause. These are evolutionary strategies, so these have evolved over a very, very long time. So we have our C3 plants, and those are most of the plants that you see in and around our environment, but there are C4 plants as well. So C4 plants, what they do is they create a 4-carbon compound rather than a 3-carbon compound. You remember that in the Calvin cycle, we make 3Pg, okay, 3Pg, that's a 3-carbon, and it turns into 3Gp when we reduce it. Well, we're making a 4-carbon molecule instead of a 3-carbon in C4 plants. A 4-carbon instead of a 3-carbon. But there's also a spatial aspect to this, and I'll explain that in just a second. We have to have a special enzyme here, and this is not found in C3 plants. It's called PEP carboxylase. Just recognize that as the enzyme that C4 needs. PEP carboxylase takes over during periods of photorespiration rather than rubisco. So a C3 plant, rubisco will participate in photorespiration. In C4, though, PEP carboxylase will take over. It can even fix CO2 even when CO2 is low. It's one of the really interesting enzymes. But here's the spatial aspect of it. You see how these different cells are arranged? What happens is you have light cycle stuff going on in one cell and Calvin cycle going on in another. It's a way of protecting the use of water for only when you need it. So you're creating a 4-carbon compound, and you are spatially separating the light dependent and the light independent reactions. So up here on the left, we have a comparison of the structures of a C3 plant that's only going to make 3Pg and a C4 plant that uses PEP carboxylase. In the C3 plant, everything goes on inside the thylakoid of one cell type, the mesophyll cell. But in a C4 plant, you have the light-dependent reactions going on in the mesophyll cell, and then you move those products over to this other cell called a bundle sheath cell that will allow the Calvin cycle to occur. And so here are some, and that's the point, that's the major point that I want to make. So make sure you understand that it's PEP carboxylase that's involved. It takes over from rubisco, that it will help protect the plant, but also that it's a spatial organization of photosynthesis. And like we show here, corn is a C4 plant. You can do much better in drier and hard and warmer conditions than many C3 plants. If you've ever been walking around the sidewalk or on campus or on a cobbled street like so, you might see these little plants right here. These are C4 plants. Another really important crop that's a C4 crop is sugarcane. All of these utilize this spatial organization of photosynthesis, the PEP carboxylase enzyme, in order to protect from particularly warm and dry situations. We've got this figure here, and it looks a little bit complicated. Just for fun, notice that here's oxaloacetate, which is a molecule that we used to in the citric acid cycle to make citric acid, if you recall. But in any case, remember, it's PEP carboxylase is the primary thing, takes over for rubisco. But here's the deal. The real deal is that we're using spatial compartmentation to conserve water. That the light-dependent reactions require water, but we want to keep doing the Calvin cycle if it's possible. So why not, shut this cell down and you can keep Calvin cycle going on over here, and that would mean that you wouldn't need additional water for those light reactions. So that's C4 for you, the C4 pathway. Another evolutionary strategy that has evolved is using this molecule called CAM. Crassylation acid. I'm never going to ask you to pronounce that, and I don't pronounce it very much. But CAM is another way of fixing carbon that doesn't require a whole lot of water, requires hardly any. And that's why you see CAM plants in the hottest and driest environments like cacti in the desert. Or if you have an aloe plant, though that aloe plant is a CAM plant. CAM uses a temporal separation of steps. A temporal. If C4 is a spatial strategy for managing water, CAM is a temporal strategy. They will only open their stomata at night when we're closer to the dew point, or hopefully at or below the dew point, in order to absorb gases and or obtain water. So the light-dependent reactions will occur with CO2. We're going to fix CO2. It's just that we're going to use the CO2 that they've collected at night, not during the day. So they close during the day, and any residual CO2 or from the CO2 released from metabolic processes and is then used in the Calvin cycle. So it's closing those stomata, so you don't have free gas exchange during the day, but and any CO2 that's there is already in the plant and being released or was absorbed at night. This is a temporal separation to conserve water. By not allowing gas in, you don't need water to do the light-dependent part of the reactions, or you do those light-dependent parts at a very, very low rate. And so here is our C4. It's a spatial way of protecting the plants. And we've got sugarcane here. That's an example of it. And then a CAM plant that's very popular with humans is pineapple. CAM actually uses a temporal separation rather than a spatial separation. We're using 2 cells in the C4. We're using 1 cell in the CAM, but it's a temporal separation rather than a spatial one. What we are most concerned about, and part of the reason why we would want to understand these sort of photosynthetic protective properties, is that we are seeing environmental change, especially over the last 150 years since the late 1800s in the Industrial Revolution. We have seen carbon levels spike in a very, very short period of time. Many climate doubters and naysayers will say, well, we've seen changes of great extent over thousands or millions of years. That's true, but it happened relatively slowly. For instance, we can also travel at thousands of miles per hour. Like, for instance, we can launch you off in a rocket and accelerate you to over 14,500 miles per hour. But you can't do that in 2 seconds. Any more that you could be going 50 miles an hour in a car and then suddenly stop. You wouldn't be able to withstand that change. And that's sort of what we're seeing here. Is we're seeing environmental whiplash is probably a better way to put it. Yes, we can withstand change if it's gradual, but when it's abrupt and acute, that's the problem. And so if we have levels of CO2 that are changing, that may affect C3, C4 plants and CAM plants differently, causing changes in the abundance of these species, especially so if they are food crops. They are relatively unpredictable. We can have some chance of predicting, but there are always additional variables that can lead to unexpected consequences, emergent properties. And we have just one more section here, which I won't spend a ton of time on, but you should hopefully have a sense of why photosynthesis is so preeminently important. The energy flow that's involved. We're using energy to bump up electrons, and that allows us to create chemical bonds and complex compounds. So we supply that chemical energy. So we make sugars that can then can be used to create ATP and carbon skeletons for organic molecules. We can also store that for future need and for during periods of deficit or non-ideal photosynthetic times. And we're also creating oxygen that we need, that most animals need in order to survive. So make sure you have these processes. You understand each and every one of the roles of these words. Walk yourself through the light reactions, photosystem 2, photosystem 1, what we're creating and where it goes. That first step, RUBP, created into 3PG using Rubisco, which is not shown here. G3P can be used to make glucose or sucrose or starch and that sort of thing. Remember what the names of these guys are, P680, P700. When P680 loses its electrons to this acceptor, it's going to rip electrons off of water. P700 absorbs electrons from this cytochrome complex. Remember, we have cyclic electron flow, in addition to linear electron flow. That first step, the fixation step, then we reduce, and then we regenerate. We regenerate after we have lost a 3-carbon sugar. So I put this in here just to show you this is how you might want to take a picture and try to write stuff in. And I think the writing aspect is a very, very powerful tool. In one of my other classes, I say, hey, you can use all the notes you want as long as they're handwritten. The act of writing helps you organize, helps you learn. This is a lovely thought experiment showing that showing the role of the proton motive force in the thylakoid. So if we isolate thylakoids, which are normally kept in a standard pH of 7, and we put it in a pH of 4, we don't get production of ATP. Well, why is that? That's because we've lost our proton motive force. We haven't denatured the enzymes. We've lost our proton motive force. There's no gradient for the protons. But if we take these and if we take these and we put them in a pH of 8, now we can make ATP because the proton motive force has been restored. That's what's shown here. If we have a pH of 4 inside the thylakoid, if you had an external pH of 4, this would never work because there's no force. But we have a pH of 8 in this situation. We're given that, so there's a 10,000-fold difference, 10 to the 4, okay? 10 to the 4 difference in concentration of hydrogens. And so we now have a proton motive force, and we're going to start making ATP. Just put the pieces together. So I've left this in here one more time. I think you've seen this enough in this chapter. But it is always good to practice and always good to try to remember what each of these pieces is doing because we've talked about all of them. And that's it for photosynthesis. I leave you with this interesting picture here. It shows the heartbeat of the world, if you will. We call it the heartbeat of the world. It shows you the relative amount of photosynthesis based on relative size. So in the summertime, you get a lot of photosynthesis, and the countries are depicted as bigger during that time. And during the winter, when the temperatures drop and the sunlight angle changes a little bit, it's a little bit lower in the sky, so photosynthesis slows down. So you can see the rise and fall pretty pronounced up here in Asia. The change in the photosynthetic rate. And so somebody created this, and now we have the heartbeat of the world. That's just how important photosynthesis is and why we should never take it for granted. Well, thank you for your time. I hope you've learned something in these lectures, and I will talk to you soon in our next chapter. See ya.